today's speaker is David Baker from the University of Washington. What we'll do tonight, I think, that worked quite well last time is David will give his talk. People, please drop in questions of any type in the chat. And then David, what we did last time is the speakers just read the questions out of the chat. I think that's the easiest way to do it rather than me reading them, because there's massive comprehension issues there with me having to understand what the question means. Yeah. What I'll add to that is if you have asked a question and David's answering it and you want to get more information, we've got plenty of time today, so you can unmute yourself and have a conversation and do that. That's absolutely fine. And we're not going to police the questions or anything like that, so we're not going to prioritize anything. We'll just do them in chronological order, and that should be fine. And then at the end, I think David's talked about having a discussion. I think we'll do that based around the framework of the questions that we originally asked people to fill out on the Google Sheet related to molecular machines, directions of research in this area. And with the extra time that we have today, that'll be quite useful. And then we'll lead eventually into what Alison mentioned about. We're talking about the bountied brainstorm. Is that how you've described it? I can never recall. It's alliterated, which is lovely. So that'll lead quite nicely into that. And then we'll move on to gather afterwards. So I've done enough talking. So David, the floor is yours.
Yeah, and let's see, like I said, oh no, it's the host disabled screen sharing. Maybe it'll just be a conversation.
No, you have full power now. So you'll.
Oh, I have full power. Oh my God! I hope I use it wisely. Yeah. Okay. Here we go. So let's see. Sorry. View present. So does that work?
Mm-hmm.
Okay. So right. So I'm going to talk about de novo protein design. I'll give an intro, and then maybe I'll give you some choices I'll give you a menu of what to choose from. So the way that we've been thinking about protein design for many years, actually up until very recently, is really based on the principle that the folded states of proteins are likely global energy minima for their sequences. So each protein has a unique amino acid sequence, and that amino acid sequence will have a lowest free energy state, which depends on that sequence. So in this picture, if you're trying to predict the structure of a protein from its sequence, you need some way of computing energies of all the of interatomic interactions. You need to sample through all the different possible structures, and you search for the one that has the lowest energy. So in this talk, though, I'm not going to be talking about protein structure prediction. I'm going to be talking about protein design. So we're making up brand new sequences that will fold up into new structures. And here we kind of do the opposite. You make up your structure somehow, and I'll give you lots of examples of that. You make up your backbone, and then you have to find an amino acid sequence whose lowest energy state is that backbone. So so over the years, we've been developing a program called Rosetta. And so what this does is it calculates the energies of any particular configuration of a protein, and it samples through the space of possible structures and the space of possible sequences. And that's how we've been approaching protein structure prediction and protein design. So very recently, deep learning is infiltrating quickly. And so we are starting to use deep learning for both of these problems. I can comment on that if there's interest. So the problem that we've really been focusing on for the last 5 or more years is the problem of building proteins completely from first principles. So sequence space is really, really big. Even if you just use the 20 naturally occurring amino acids, there are, for 100 residue protein, you have an astronomical number of possible sequences. And the number of proteins which have existed on Earth is just a tiny, tiny fraction of this. So there's this enormous space of possible proteins that evolution hasn't explored. So that's kind of depicted in this gray box here. And so in red would be not to scale, but naturally occurring sequences which clump into families because of as evolution proceeds incrementally. And almost all protein engineering has consisted of making small changes to proteins which already exist, which we affectionately refer to as Neanderthal protein design. And what we're doing is trying to build proteins completely from scratch that are unrelated to any proteins of known structure. So we've been doing a lot of this over the last 5 years, and so let's see. So what I have here, and this maybe I should just skip this entire section. Maybe I'll just summarize in words on this slide. So for the last year, obviously, we have been, like the rest of the world, very preoccupied with the coronavirus. And so we used this sort of de novo protein design to design brand new proteins that block the virus from entering cells, that's antivirals, that sense the virus, it's diagnostics, and induce an immune response against the virus. And I could sort of run through this quickly, or I could just maybe I don't know. I'll just give you to sort of make this protein design problem more concrete. I'll just go through the first one. How about that? So as you know, the coronavirus gets into cells by way of this ACE2 receptor protein, and so this is sort of a depiction of a classic protein design problem. You're given the structure of a target, like the spike protein of the coronavirus, and the challenge is to design proteins which bind to it. So here we generated in silico very large numbers of virtual protein structures, and then docked them against this surface and designed the interface for really high affinity interactions. And the blue one here and the orange one here are some of the solutions that came out from this. And they're kind of remarkable proteins. We've made them in the lab. This one is only 55 amino acids, so it's very short for a naturally occurring protein. It's rock stable. It doesn't melt. And then, of course, there's a question of did this calculation actually get the answer right for the right reason? And so we can. What we did was to solve the structure of how this protein actually binds to the coronavirus by cryo-EM in collaboration with collaborators here at UW. And so this is the spike trimer, and this little thing is our designed protein. And so you can see 3 of them bind. One of these binds to each RBD on the spike. And we can now compare this to the model that we had made with Rosetta when we were designing it. Remember, I told you we were making up designing a protein that would bind tightly. And what's really quite remarkable is that the structure of the design protein is nearly identical to the actual experimental structure. So in this case, the sequence we designed really folds up to exactly the structure we wanted. And beyond that, it actually makes almost exactly the interactions that we were aiming for with the target, with the coronavirus, which is down here. So the methods are good on a favorable day to actually design the sequence completely from scratch that folds up to a new structure very precisely that binds a target in exactly the way you've designed. We have been using this procedure to make small proteins like this that bind to many proteins on the surface of cells, trying to make new types of signaling related therapeutic proteins. So I'm going to kind of skip through all of this data, except it just basically shows that these proteins actually do work therapeutically. They prevent animals from getting sick from the virus. And I think I will just go through. So this is probably the most relevant slide. So there's all these escape variants that people are worried about now. And we by the actually, maybe I'll go back one slide if I can able to do that. Maybe I'm not able to do that. But anyway, we can make versions of these that that are trivalent, that match the 3 RBDs on the spike, and these versions neutralize all of the variant virus, South Africa, UK, Brazil. And so we're pretty excited about these. We can make them in large amounts in bacteria, and they're headed for clinical trials, which is kind of cool. These are just completely computer generated proteins. So that's the antiviral. And maybe this is now maybe a little bit more what you're interested in. This is like design of a molecular device. So we wanted to use this for sensing. And so the idea was we could design a system which had 2 states shown here. And there'd be this off state and an on state. And the presence of the virus, and these two would be in thermodynamic equilibrium, and the presence of the virus would pull the equilibrium to the on state. And that's sort of depicted here. And the way that, and so when this thing opens up, it allows reconstitution of an enzyme called luciferase, which generates light. And this works really well. So if you take this molecular device and you add, it doesn't emit light, but you add the virus or the RBD or the spike, and you get this rapid increase in luminescence. And we're now, so we can design these multi-state devices that are emit light in one state. And we've actually, maybe I'll just skip through this part. We've used this to make, basically use this method to make devices that sense a wide variety of different compounds. And so, for example, this is troponin, which is involved in heart attacks. This is botulinum toxin. And in each case, we take all these sensors, we add the compound, only the sensor that was designed to bind, to example, for botulinum toxin binds to botulinum toxin. On the vaccine side, maybe I'll just talk about this. So we've been designing methods for making self-assembling nanomaterials. And this was one of the very first examples where we take a protein with 5-fold symmetry and one with 3-fold symmetry, and we design interfaces between them such that the 5-fold and the 3-fold sit on the 5-folds and 3-fold axes of an icosahedron. And using this approach, we've been able to make many different types of very homogeneous nanoparticles. So you make the two proteins, mix them together, and they form these beautiful assemblies. So my colleague Neil King has been making vaccines out of these by putting the RBD on the nanoparticle. And these turn out to be extremely potent vaccines. They're in clinical trials now, and they seem to be considerably more potent than what's in the mRNA vaccines. So I think I'll skip that. So I'll give you now a few examples, and then I'm going to get to molecular machines, which is what I'll spend most of my time on. So this week's issue of Science has another cool example of a design nanostructure. So here, this yellow, what we realized is that when for these symmetric materials, the basic principle is we match axes of symmetry of the protein building block with a point group or space group symmetry operators. And so in this case, we have a pentamer that sits on the 5-fold axis of an icosahedron. And on the 2-fold axis, we place antibodies. And the nice thing about antibodies is that people made antibodies to just about anything, everything. And so by using the antibody 2-fold axis, we basically have it's the constant part of antibodies have a constant part, and the variable part is what gives them all their different binding activities. And the constant part essentially becomes structural part of the nanoparticle. And so the point of this was we could really unite form and function, because we could use the antibody itself as sort of a structural building block of the material, and then have the sort of the business ends open for interacting with the target. And we made things with many different symmetries. So here's a dihedral version with 2 antibodies. Here's a this one I think is I can't see whether it's cubic or tetrahedral. So you make different by changing the arrangement of these arms and how they interact with the antibody. We could make things that form different types of nanomaterials. And here's sort of a list of them all.
It reminds me of the protein version of Ned Siemens' old work with DNA.
That's right. It's exactly that. So, and a lot of what we've done here is just sort of it's sort of following in Ned's footprints, but footsteps, but with proteins. Exactly. And then what's interesting, the nice thing about proteins, they have all these functions. So when we assemble antibodies into these nanocages, they have all sorts of new biological activities, which I think I'll skip because, okay, so here's another example of a design nanomaterial. So here we've taken 2 protein building blocks, one that has D3 symmetry, one that has D2 symmetry, and designed interactions between them that drive formation, that are predicted to drive formation of a hexagonal lattice. And what's really cool is when you mix these two proteins together, you get this really, really extensive, very regular hexagonal lattice. And this is electron micrograph. And if you look at it, if you average, you can actually, it's quite close to the design model. Here's one component and here's the other component. So if you're ever in need of protein chainmail, let me know. What we've been doing with this is attaching binding domains for cell surface receptors, and we get the cells to undergo all sorts of interesting transitions. But obviously, sort of one idea is self-assembling circuitry. A lot of, and I know this has been a theme with the Foresight Institute for a long time. A lot of manufacturing is top down, and here we can proceed bottom up, because everything I've shown you, those nano cages of all the different shorts and these kind of hexagonal lattices, these aren't patterned from the top down. These are made by programming very specific interactions between atoms. We can actually use these kind of devices I described earlier to do calculations on the surface of cells. And so, like, one of the challenges in cancer therapy is, could you have little machines that go in and kill the tumor cells, but leave normal cells fine, untouched? And that's a hard problem, because cancer cells don't often differ very much from normal cells. But if you could do calculations in the body on the surface of cells, then you could make more subtle discriminations. So here this is sort of a diagram of how we discriminate cells that do AND logic on the surface of cells. So discriminate cells that have two markers on their surface as opposed to just one or the other. And this really works. I don't think I'll take you through the details, but we can actually do one A and B, not C logic. So the basic way, roughly speaking, how this works is we have sort of a closed, one component which is in a closed state. If another component gets brought to it, it opens it up and we get a signal, which in this case, CAR T cells can use to kill the cells. But then we can also have a third component that we can target, that gets targeted if there's a third marker present, say a healthy cell marker present on the cells, and that shuts down this whole thing. And so you turn it off. So you only get response on the cells that have markers 1 and 2, but not markers 3. So in terms of building devices, one of the really interesting types of proteins in biology are channels, things that allow ions to go through. So of course, we've been interested in building those from scratch. So here are crystal structures of different types of cylindrical pores we've developed. And we can actually incorporate these into membranes. And when we do, it turns out they are very specific channels. So this one, for example, is a specific potassium channel. So what's kind of interesting about this is people have been arguing about why potassium channels in nature are specific for potassium for a long time. And here you can just sort of, and so a lot of biology is descriptive. You observe things and then you try and build up explanations for it. But here we can just sort of prove by construction. And so here we built something from fairly simple principles and ended up with a fairly specific potassium channel. I think I'll skip this one. We've also designed larger pores here that allow larger molecules to go through. And then so what I've described so far are helical transmembrane proteins that have rings of alpha helices. But some of the really interesting transmembrane proteins that are, for example, used in single molecule DNA sequencing are beta barrels and that sit in the membrane. And we've recently learned how to design these proteins from first principles. And so now we are designing a series of proteins that look like this with larger and larger pores in that we think will be able to functionalize for all kinds of molecular filtration processes and in addition, more sort of new approaches to single molecule analysis, because you can look at the same way that single molecule DNA sequencing works is you can look at changes in conductance as things go through the pores. We have a game that's sort of an adjunct to what we do, where sort of just people in the general public can go in and design proteins, and they can pull the proteins around, sort of as illustrated here. You can change the amino acid sequence, and we've been been testing a lot of the designs in the lab and getting a lot of interesting ideas from all the very smart people out there who aren't professional scientists. Okay, so here, this is I'm going to go in a little more detail here. This is now actually our steps towards designing molecular machines. And this is the work of a super talented postdoc in the group, Alexis Courbet. And these are his slides, which I showed, which I stole as well. You all recognize the quote at the top. And so his idea was to design machines with out of rings and basically rings and axles, and try and assemble rotary systems at the protein level. So this is kind of at the mechanical level. He's trying to assemble things like this that have like kind of an end, a cap on the end. Here's an axle, and then here's a rotor that spins on the axle. So the basic idea is to design these parts, the rings and the axles, then assemble the ring on the axle, and then incorporate chemical reaction at this interface to actually drive rotation. I'll show you where he is on this. As you can tell, he's a little more colorful in his slide construction than I normally am. So the first class of materials are these axles, which are basically rods that generally have something that we built, that Alexis built something on the end that sort of hold the rotor on, the ring on. And these are different types of constructions. These are electron density maps, and he's got quite a few different shapes. And what's interesting is that they have different symmetries, so this matching or mismatching of symmetry will be an interesting thing as I go through. He can extend these to make it easier to follow by imaging. So those are the rods, and then here's the rings, and he has got quite a few different flavors of rings. These are all, like everything in my talk so far, these are all completely de novo designed proteins. So we've got these rings that are structurally validated, these rods that are structurally validated, and now here's just some more structures of the rings, just so you can see what the rings and rods. These are the ones that he has the highest resolution structures of. So you can see we want to put these various types of rings on these different types of rods. And I actually thought that was going to be pretty hard, but it turned out that Alexis was able to thread the rings on the axles pretty straightforwardly by doing some primarily incorporating electrostatics between the ring and the axle. And here are and we can play tricks like using pH or disulfide reduction to actually assemble and trap the rings around the rod. So these are some of the systems that Alexis made now. So there are ones where the symmetry matches. So both the axle and the rotor have C3 symmetry. And then there are others, these other ones here, where the symmetries don't match at all. So the axle, for example, here has D3 symmetry, but the rotor has C5 symmetry. And what you see here are actually cryo-EM maps of the rings, the rotor, the rings assembled on the axle. And here you see some views of these. Now, what Alexis has done is to calculate the energy landscapes in Rosetta, basically just doing that. There's 1 degree of freedom as the ring rotates. And you can see the landscapes look really different. As you might expect, when there's symmetry matched, you have 3 pronounced minima, because there should be 3 rotationally identical states. Whereas when you have symmetry mismatches, for example, D8 on C4 or D3 on C5, you get much more rugged landscapes. And here's an example of one of these. This is the one that was on the left in my previous slide. And so by cryo-EM, Alexis has actually solved different states of this system, and you can kind of see them here. So they are what we can distinguish them because there are 2 rings that are mounted on on the ring, and they have these things emanating from them. So you can see here they're more eclipsed, and here they're more staggered. And it's kind of neat. They correlate to minima in this rotational energy landscape that Alexey has mapped out. So we can design these systems that have multiple rotational states. So now the question is, can we get them to undergo directed motion? So the way that Alexey has approached this is to build in catalytic sites. Many years ago, we started doing enzyme design with Ken Hauk, who I was happy to see here. And so this is a reaction that Ken will recognize, the retroaldase reaction. We've put a very simple site in it, this interface between the ring and the rotor. What we know at this stage is that this is an active enzyme. There's so the enzyme turns over, and we can also chemically modify this site with different chemicals that are sort of like suicide substrates. Here's a picture of how the substrate fits into this binding site. And whoops, sorry. So here are some of the compounds. So that we get enzymatic activity, and so we have so obviously what we want to know is what happens when we add the substrate. So we're collaborating with Zev Bryant's group at Stanford, and they get these very tantalizing they basically put a gold bead on the end. And of course, what we want to see is rotational motion. And so this is sort of angular motion over time. But there's a serious data selection bias here. So we don't really have definitive data yet on whether we have sort of unidirectional rotation, but we're working hard on that now. These are just different trajectories Zev's group is getting in their single molecule experiments. That's pretty much where we are now with the design of these rotational systems. I should say we're also designing systems. I didn't show it in this talk, but we've designed a variety of protein one-dimensional fibers using the same self-assembly systems, and we're now using sort of similar principles to try and design walkers that walk along the fibers. So that's probably a good place for me to start. I have a little bit on deep learning as opposed to as applied to protein design, but maybe that may be of less interest here. So just to summarize, I think we understand a lot about protein folding and assembly, and now we can really start building a whole new world of functional proteins, which is very exciting. And just to quickly acknowledge the people who did the work, Longjin and Brian did the design of those the mini proteins that block coronavirus. We have a lot of collaborators for all the animal data. The molecular devices that sense the virus is work of Alfredo and Andy. The self-assembling nanocages, those antibody nanocages, Robbie Devine's work. That two-dimensional sheet is designed by Ariel Ben Sasson. The membrane proteins that the synthetic potassium channel, work of Chun-Fu and Pei Long, and Anastasia designed the beta barrel. The systems for doing logic inside cells, work by Mark and Scott and Jillian. Folded is the work of Brian Kovnik and a number of other people really all around the world now. And Alexey Corbet is really the kind of the genius behind the rotary motor stuff. And I didn't really talk about the protein hallucination. So why don't we go to discussion mode here? Actually, I guess I can check. I'm supposed to check the questions. Is that in the chat?
We do have a few questions.
I don't see.
Well, we have a few hand raises. Let's see. We have Creon, and Creon, maybe say one or two words about you, and then we'll move on.
Okay. I can also talk for 2 minutes at some point about the deep learning stuff if people want to hear.
Oh, I'm sure they want to hear about that. Yeah, I'm Creon. I'm a longtime Foresight person and worked in nanotechnology and quantum chemistry years ago. And lovely, beautiful work. And I knew Ned Seaman and all those guys back in the day. Anyway, so I'd like to know, maybe one level down, if you don't mind, like under the hood of some of your modeling, to what degree is it molecular mechanics? To what degree is it electronic structure? You know, all the complicated stuff, like what about the water?
Yeah.
You know, what about the reactions? Is it just shapes? Is it more than that? Or is it all like machine learned concepts where you don't have to worry about physics? If you know
it's a really good question. So it's basically molecular mechanics with basically the main difference between the Rosetta force field and a traditional molecular mechanics force field is that we treat the hydrogen bonding orientation dependence explicitly, because you need these hydrogen bonds give you a lot of the precision and the orientation control. And when you have two surfaces coming together, you have to make all the hydrogen bonds, those groups were making interactions with water before. So basically, but the functional form, it's basically a pairwise additive sum, largely pairwise additive sum over atom-atom interactions, the Rosetta force field.
Okay,
so,
but wait a second. That's great. Thank you for sharing that. That's amazing. But that's not going to make, you know, a sodium channel or a motor, right? There's more electronics going on in there.
Yes, you're absolutely right. You're absolutely right. And for example, the enzyme work we did with Ken, you know, obviously that's clearly gets into quantum mechanics. So, but there is an issue of control and what control we have and what control we don't have. So, so I think so you could sort of think about it as, so for the potassium channel, for example, we designed structures using sort of this force field I described that had a cylindrical aperture that was about the right size for potassium channel passage, but we did not explicitly model before the fact the dynamics of potassium traversal. Likewise, in the molecular machines, those rotary machines, you know, so it's a sort of a combination of the sort of atomic level design and then shape reasoning, right? So Alex, he designed the rings and the rods, but in terms of precise modeling and we had these after the fact energy landscapes, but and then I showed you we designed catalytic sites in there, but we're not computing in advance what we're not choosing the catalytic site based on what would maximally power rotation. It's much more heuristic than that. We put it at the binding site. So there's a combination of sort of real atomic detail at the base design with sort of hypothesis, build, design, build, test at the at sort of the more meta level, because you know, these our calculations just aren't accurate enough at this point to work out exactly how to design this molecular rotary machine.
Okay, one last question, follow-up, is that this is beautiful, what you've described, this sort of multi-level analysis with molecular mechanics at one level and kind of human heuristics at the other level. Tell me a little bit, if you will, about the feedback that's happening. Like, presumably the mechanics are informing your heuristics, and then maybe your heuristics inform the mechanics.
Yeah, well, there's a lot of feedback, and part of it comes from something, in a sense, my talk was probably the most misleading thing you've ever heard, because I showed you all these beautiful things that work, but 95% of what we make doesn't work. And so we get a lot, and so typically there's a lot of different failure modes. So basically what we're doing is we're doing these design calculations. We get out a protein structure, which has a particular designed amino acid sequence. We make a synthetic gene that encodes that amino acid sequence. We put it into bacteria. The bacteria make the protein, and some reasonable fraction of the time, they simply don't want to make the protein at all. So there's no protein made. Other times they make the protein, but it's just, turns into gook at the bottom of the tube. And other times it makes a perfectly well-behaved protein, but it doesn't do what we want. So we get a lot of feedback. We're getting a lot of data back on what works and what doesn't. And so there's definitely been a lot of iteration. So we can go back to the force field, look at try and figure out what's wrong, improve the parameterization and so forth. Also, our understanding of how to approach problems is evolving as well. Really what you described is exactly it. This very detailed physical model with heuristics sitting on top of it. But maybe what I should do is just show a couple more slides on the deep learning stuff, and you'll see how that's or I can go through the comments more. I don't know, whatever people would prefer.
Well, there's been questions about learning, so we all say yes.
Yeah. Okay. So obviously, like I said, we're collecting a lot of data, and so there's a really interesting opportunity to use ML. Let's see. So if I don't know how I get back to my slides.
You should be able to if you're still co-host, so if you go to the bottom and where
okay.
it says share.
Okay. Sure. Yeah. Okay. Sorry. Where did it go though? I'm sorry, I'm just having a hard time finding the.
If you go to the green button where it says share screen.
Yeah, no, I know. I'm just, for some reason, it's giving me all my mail.
You have to first click on the presentation. Like you have to first search the presentation, click on it, and then you go, and then it should be the first one that pops up on the left. But if you had a different browser on before then it just shows you that browser always.
Okay.
Open your slides and don't minimize them, and then go back to Zoom.
Okay, let me try that. I thought I just tried that, but it didn't work. So if I'm in presenter mode, if I am in presenter mode, I'm just somehow, it's giving me 100 zillion mail messages. Why the hell is it doing that?
Well, show us them.
Yeah, you know, I mean, okay, let me just try. I'm going to try exactly what was suggested. So I am in present. So I go to presenter mode, but then how do I get from there to back to the Zoom window to?
Or just click on this.
You don't have to be in presenter mode. Just bring that up as the last document. Don't minimize it, and then go back and share screen, and you should see it.
Okay. Yeah, it should really be there. I just don't know why it's not. Oh, here it is. Here it is. Okay, sorry. I don't know why I wasn't finding it before. Okay. So
that always happens when you talk about AI.
Yeah. Okay. So so we've been playing around with so basically the prelude to this is we've been working on structure prediction is a big industry in my group. So we've been working for several years on methods for predicting protein structure from amino acid sequence. And we, like everybody, have been DeepMind has been doing great work on this. And so we basically, I think we're sort of the people who actually make some of their ideas available to the world because, we basically try and build on what they do. So I'll talk about very briefly, basically this idea of deep network hallucination. So here's protein structure prediction. You have an amino acid sequence, you predict the structure, you get some kind of, well, what we actually predict is, in this instantation, is a distance map, distances between all these different pairs of residues. And what we can then do in the same way that if you have a deep network that recognizes images of cats on the internet, that you can sort of feed it noise and then optimize the noise so that the network really thinks it's a cat it's looking at. That way you can use to sort of hallucinate brand new images of cats. Well, we can use the same idea with proteins. So start off with random sequences, predict their structures. They don't look like proteins, but then we can optimize the sequence such that the network really thinks it's a protein. And so this is a residue by residue contact map. And if we do this optimization process, at the end of the day, we get these very featured contact maps. So this is residue by residue, and you get a black dot if those two residues are close in contact. And these these correspond to protein structures. And when we do this, starting from different random number of seeds, we get lots and lots and lots of things out. Lots and lots of proteins out. They look like proteins. They're not related to any naturally occurring protein. And Rosetta, which in this molecular mechanics model, predicts that they actually fold up to the hallucinated structure. We've made a whole bunch of these, and they actually fold in the lab. And we've solved structures now of 3 of them. And actually, these hallucinated sequences fold to the hallucinated structures, which is pretty neat. One of the things that this enables is sort of a shortcoming with our sort of physically based method for protein design is we were always, when we were optimizing the sequences, we were optimizing for trying to minimize the energy of trying to find the lowest energy sequence for a structure. So that's kind of depicted here. This was everything I described so far, but there could have been off-target structures that were competing that we didn't know about till the end of the day when we tried to search the landscape. But with this approach I'm describing, we can actually explicitly maximize the probability of the desired structure as opposed to other possible structures, because we're essentially using the network to predict a probability distribution over all possible structures.
What was the difference what was the difference that took you from the first state to the second state?
Yeah, so that's a good question. So in this molecular mechanics model, right, you have the interactions between atoms, and it's very nearsighted, right? It just sees the structure that you have at the constellation of atoms you have at any given point. So if you're trying to optimize the sequence to find the very low energy sequence, you're doing this combinatorial search in sequence space, trying to get very low energy interactions. The network instead is reasoning over the probability distributions of distances between all pairs of residues. So as you change the sequence, it's updating all those probability distributions. And so you're essentially, yeah, it's like, you know, the deep learning thing essentially knows about the partition function implicitly when it's doing its probability estimation. Okay, so the last thing, and then I'm going to shut up, is we can start designing things that have function by constraining parts of the protein to have shapes that are relevant for protein function. And just to give you one example, so here's a big protein that's part of the complement cascade, it's called. It's this big protein. It's got this business end down here in green, which binds target here. What we can do is take this green part out, say we want a protein that folds up to a protein that has this structure. We want everything else to be free. And then the network will just hallucinate different proteins that have this region. And we can do the same thing like with an enzyme active site. We can say, okay, we want this part to be fixed, and then the rest of it can be whatever the network thinks is best. It's like fixing the cat's ears and then asking the network to come up with the rest of a cat that would have those ears. Yeah, so this has been the work of really talented people who are listed here. Yeah, so that's kind of the deep learning for sort of new protein design. And then there's also the very exciting area. We're generating all this data from all of our failures and using that to iteratively improve the model. Now, so I'll stop sharing now. So there's now this, you can totally appreciate the issue though. So what I always thought we were going to be doing is we're collecting this data to improve the physical model, you know, the parameters, the functional forms. But now, you know, in these deep learning models, there are no functional forms and there's millions of parameters, and, you know, they work, but and I say we're at this stage now where there's sort of this tension between the deep learning models in my group and development and then Rosetta. And right now we're kind of going back and forth, but it's hard for me to predict exactly how that's going to evolve. It's going to be very interesting. Oh, I see a question. I'd love to hear more about the self-assembled 3D porous materials.
I'll say before we get to that, David, sorry to cut. I mean, Dirk Trainer had his hand up for a while, so we'll have him ask his question first, which I think came in before the questions in the chat did. Oh, and sure. I told him he could ask it, so that's what we'll do. Sorry.
No, in any order is fine.
Hi, David. This is Dirk from NYU. David, back to that rotary machine, which I found absolutely cool. Reminds me a little bit of T. Ross Kelly's chemically powered rotor. I don't know whether you come across that. So how is yours powered exactly? If I understand this correctly, this is a retro-aldol reaction, right? Which should have a positive delta H. Is this purely entropically driven, or what is the power stroke in this machine?
Yeah, that's an excellent question. And so what I can say at this point is we have not, so what we can do is we can assemble these systems, and they clearly are undergoing rotational diffusion, but we have not convincingly demonstrated directed motion. So there's, it's basically, this comes back to the previous comment. So we are still trying to characterize, you can imagine it's a bit of a tricky problem. You need to do single molecule fluorescence or some other measurements to see. So basically we've been faced actually for over a year now with just technical problems and how to really monitor what's going on. So, so the idea ultimately would be some sort of Brownian ratchet, right? That the binding of the substrate changes the energy landscape. And then there's actually, after the hydrolysis, there is, you remain with the product bound, then that comes off. So the hope is that we can sort of deform the rotational energy landscape in such a way as to get directed motion. But we have not convincingly demonstrated that now. And actually, if anybody has ideas about how to characterize these systems experimentally, you know, we need help.
That would be really cool to run it through some form of decarboxylation by making a gas, right? Or something like that, or some something along these lines.
So I think that was the only question with a hand up. So David, you can now go into the chat. Sorry to have cut you off with the
so
previous
as far as
one.
3D porous materials, we're working now. So those arrays get to be tens of microns long. I think they would get bigger at some point. You start using up all the protein. We actually now can design 3-dimensional crystals, and we actually have been designing hydrogels that look like non-Newtonian fluids. It's very interesting. So I would say there's not really a limit on the size. I mean, because these are all symmetric systems. It's really just they're limited by the amount of material that you produce.
And then Tad's got his hand up, so I don't know if you want to.
Oh, for protein logic, what's the limit on number and accuracy amount of logic?
Okay. Thanks. Hi, David. I enjoyed this discussion. So I have a couple of questions on the protein logic that you described. And so first I'm wondering what's the limit on the number and the accuracy of the Boolean operations that these devices can perform on the surface of a cell? In particular, if you start having a lot of logic you'd like to do the devices get in each other's way? So you have to kind of space them apart, and then maybe there's errors in what they implement. And the second piece of my question, I was wondering whether you can include delays in this logic so that you could detect there's a receptor 1 that happens, and then a little bit later it's number 2 as opposed to 2 in the order of 2 and 1. So can there be delays in that? It's a little bit more complicated than just Boolean logic.
Yeah, no, that's a really good question. I would say this is a rapidly evolving area. And I think there are issues about crowding that would come up ultimately. There's also, I kind of glossed over it, but the way that we're targeting the logic to specific cells is through domains that bind to proteins that are expressed on the surface of those cells, and we need those to be present at a high enough level. So right now we're kind of trying to optimize those sorts of things. We're also designing synthetic receptor systems so that you can control cells from the outside by and there you can get delays. We're building integrating devices that sit inside the cell that would sense the activation of multiple different receptors. And so that's sort of the way that we're trying to get at sort of timing more from the cell, trying to put these design devices into cells.
Okay, thanks.
Yeah, how would you, what's the picture of going from machines to function? I think all of you will sympathize. I mean, so basically my experience has been whenever you can achieve a real sort of breakthrough in technology, there's always applications that you can't anticipate. And so we have not really, there are crazy things you could imagine with a rotary machine would be like cleaning up, amyloid fibrils and neurodegenerative disease, because basically if you had that, there are rotary systems in biology that untangle DNA, for example, but we really haven't delved into that much. Chaperone and nascent protein interactions, we haven't really looked at that. We're really focused on de novo protein design. And most of the proteins that I described, I'd say essentially all of them fold spontaneously. So, so that hasn't been so much of an issue. And then, hi Ken, thanks for the nice comments. A lot of the failures are due to aggregation, I would say, because a lot of these systems, they interact via some hydrophobic interactions, and so those can also lead to aggregation. So how does the design space work for flexible structures? Well, a lot of the design stuff we're doing now, I would say, is not completely flexible. We're trying to design multi-state systems with multiple discrete minima, and there you basically just have to come up with there the design part is harder because there has to be multiple independent states. And as far as designing intrinsically disordered proteins, it's very easy. That's what we call failed designs are basically intrinsically disordered. So, but we also have systems that are disordered but then become ordered upon binding. Yeah, I think that's most of the questions on the list.
Yeah, I think that's caught most. I mean, Ben's question, sort of the thing that one of the questions that this group is sort of targeting is sort of speculating on where things might go in the next like 5, 15, 30 years. And so, I mean, obviously there was like very sort of specific connection that was asked there about dysfunction. But do you have any, like, because I mean, as far as I'm concerned, the sort of stuff you do is basically magic, as far as I'm concerned.
I would say patterning. I would say the overall goal is like really patterning matter at the atomic scale. So a lot of the this principle of self-assembly, we're able to pattern things, you know, with these, you know, hexagonal lattices, nano cages, different point group symmetries. But if you want to pattern more extensively and asymmetrically, then I think that's where things like the motors come in, like the walkers, because you could now start moving things around. So I think that's one way to think about, sort of like, suppose you wanted to assemble an asymmetric circuit, then you're going to need something that can do work and that you can control. So I think that the general form of the answer to the question, I would think about these systems that use chemical energy is actually patterning matter in highly directed ways at the atomic level.
And so that's probably, would you mean, this is me linking to the sort of questions you'd say that was an area that you feel the community should focus on as like where there's maybe a lot of high value to be had from doing that patterning in that way?
That's certainly a very general form of the problem. I don't know. I think it is hard to anticipate. You know, I think that's sort of the history of technology is they're sort of technological revolutions, and then people figure out what it's really good for after the fact. Yeah, we'd just be really happy to see a spinning motor, a unidirectionally spinning motor, or a nice walker walking along a fiber right now.
Yeah, absolutely. I mean, if there are no other questions, as we've done fairly well for time, Alison, I suppose we can probably go to your.
Yes. Well, okay, David. I mean, we still have 3 minutes of you. If you don't mind me just cascading questions out to you, and you just see which you get to.
Not at all. But I think we went through all the questions.
Okay. Well, I have
another one's just come in actually.
That we ask everyone in the.
Sorry. Oh, wait. Yeah, sure. I forgot about that. Yeah, go ahead.
Okay, well, so is there a tool or an enabling technology or something that you really, really wish you had that would significantly advance your field? Where you're like, if someone listens to this in the engineering department, make me this, I want that.
Yeah, well, like I said, we're having a hell of a time characterizing these rotary machines. So sort of measurement devices for measuring directed motion on the nanoscale, that's really a major limit right now.
Okay, and if you could ask people in this group, what do you think is like the #1 question that people in this group should have an answer to drive progress in molecular machines? Like, if you were in my position, what would you ask everyone here?
I think it is, a lot of it is measurement methods. Like, I would say develop methods for quantifying the behavior of molecular machines, because to some extent you can't really design before you have measurement. So I think I would really urge sort of thinking about, you know, how will you quantify the behavior of molecular machines as people start designing them.
All right, thank you. And then we have one from William Shee still in the question: can you measure rotary motion with a micron length lever attached to the rotor?
Yeah, that's a really good question. And let's see, so one way you could do that is if you made the arm really long, you could put a fluorescent group on the end, and then you could watch it. That was what was done for the ATP synthase. And that is, we're basically playing around with engineering ways to do it. Yeah, so that's certainly a very good idea to basically, that's sort of transferring, the length scale over what you need to measure, amplifying it by basically going out to a larger circumference circle. Yeah, so I think that's a very good idea.
Okay, and then 2 questions for 1 minute. One is, what would you recommend young people in your field to focus on now and to do? And then the other one is, what can this group to help with your work? Is there anything we could do for you after you've gracefully given us an hour of your time? What do you need?
Well, I think there's a really stimulating discussion. I mean, I think, again, if people have ideas about measurement methods, I think that would be really good. I think sort of the question about what would you do with machines once you had them? I mean, at some point, this field is going to have to make an argument for why you need them. And I think your group is very, very well poised to that. So I think that will definitely help build enthusiasm for these, because of course you need some kind of society buy-in to really do something like this. So I would say measurement methods, applications, and then you had two questions. So, and I think that was the second one. What was the other one?
What would you recommend young folks like young David?
Yeah, let's see. Well, I think one of the things I would recommend is to try and become a generalist, because there's a lot of different things coming together here. There's the physics and engineering of measurement. There's sort of the physical chemistry, biology of machine, proteins and machines. There's the sort of the computer science of deep learning. So really trying to getting a broad view. There's so many different things converging in this field. That would be my suggestion. Don't become a specialist too early.
Okay, thank you. I hope you didn't mind that I tried to fit them in. But,
no, not at all. I would have felt bad otherwise, because I just wouldn't have remembered. Let's see. There's
well, thank you very much for your time. It's really much appreciated.
Sure, of course. No, that was a pleasure. And if people have ideas, definitely send me an email, particularly about I'd say about things like measurement problems and applications.
Okay.
Yeah. Okay. Nice to talk to everyone. Yeah. Goodbye.
Thank you, David.
Sure.
Thanks.
Yeah.
Bye. And everyone else who'd like to stay on, Ted and I have a little bit of a report out. I just want to make sure that we get all the time that we have with David. But we had a meeting on Saturday on the Molecular Machines Bounty Brainstorm. So where we pay you prizes and you answer the questions, some of which I asked David now. And so the idea is that, we pay you for the expertise that we have so that we can generate a report that we can then put in front of the eyes of potential funders, some of whom have funded this group. So hopefully there are some potential that this funding will get us somewhere. And Ted has a little bit of a report out, and then for everyone else who'd afterwards just like to stay on and socialize, I'm going to open up our gather space in which you can meet other folks here. Okay, Ted, I'll hand it over to you, and I will allow you to co-host.
Okay, am I unmuted? All right, I'm pasting into the chat. Okay, there's a URL in the chat. So in the Molecular Machines group, we've been building a table, which probably all of you know about already. Let me see if I can share my screen. Let's see here, so this one here, okay. Okay, and share. Sorry, I'm new to this. Okay, so this is a table of devices. So people keep mentioning all these things that were in journal articles that do something or it demonstrates some principle. And so I wanted to collect them all in one place. Actually, Tom Schroeder suggested that we do this because I think it will have a benefit later. And so we've started on this. And we on last Saturday, we put a bunch of things in here. So all of you, it's open. Anybody who has a URL can edit. Please either take one of the articles mentioned here with nothing beside it, like this one, and go look at the article and fill in the rest of the table, or have your own idea of one of these devices that is one you know about, and make a new row for it and put it in. And we would be very grateful to have this table filled in. It's really important. Okay, good. Now I would like to, now on another subject, let me get another URL, just a moment here.
Did you want to mention our nomenclature for how to notate who put in the entry?
Yes, please. Yes. So the column on the right hand of that table says initials. And so if you put in a row or did most of the work on the row, just put your initials in there and maybe your email address, since we might not know exactly who you are. And yeah, so email address and name in that initials column would be great. Okay, back to here.
And if they did just one cell, then they would put it there.
Yes, yeah, you can put it at the end of the cell, like in brackets or something like that, if you just did one cell. Okay, I'm having trouble getting back to the chat in Zoom. Anybody want to help me here?
Yeah, I would say just close the recording. Sorry, not close the screen share. Go back.
Yes.
Into the chat.
Okay,
so we just hit escape.
Wait, I think I've got the chat button here. No. Oh, here we go. Now all right, good. And okay, and okay, so this is on another subject now. Going back to this, let's see, oops. How do I get rid of the chat?
So So he just shared with you a link in the chat.
Did that come through my link that
Yes.
was shared? Okay, good. Now over here, come on, just a second here. Okay. Oh, come on. Okay, this. All right. Can you see this thing? So on February 18th, Dean Astumian talked about a very interesting thing, and I know he's maybe actually in the talk. If he would like to say something here, that would be wonderful. The proposal is a rod with a couple of rings on it, and these rings attach A and B, and then you change the pH and bring the two rings together, and then A and B might bond to each other, which would be great, because then you could go this way and get rid of the bonded pair, and then go around and around and around, powering this thing, and you're basically forcing a chemical reaction to happen. So it's a chemical synthesizer, sort of at room temperature in a single flask without filtering. This would be I think it would be a great product, and would be the first stepping stone on a chain of of commercial activity, which would be really important. And of course, we don't know exactly how to build this. I'd like to know if any groups are working on it. I don't know yet if any groups are actually trying to do this.
Stefan just said in the chat that there are.
Okay, let's see if I can get back to the chat here. Okay, John.
I don't know, Stefan, if you would like to unmute yourself to say something.
Okay,
Okay,
good.
fine, but anyway, he shared a link.
Yes, thank you. I appreciate that, and I'll go look at that. Also, the thing that's able to make 4 different shapes of the same molecule that we've talked about before, that has the arm that swings back and forth, that must be able to release those products and get a new substrate to start on. So that so parts of this have been done, and of course the rod with the rings on it has been done. Okay, so let's see. Okay, so my main message here is that there's a little paper about it. That the URL I put in the chat leads to this paper here. So if you're interested, look at that. And if this is all incredibly inappropriate, you can tell me. But I think we're really onto something here. And Dean Astumian has really opened Pandora's box, I think, here. So that's pretty cool.